Welcome to Easy Microbiology.
Good morning, everyone. Today we will be exploring a very important topic. Campylobacter jejuni. We will be looking into its habitat, morphology, cultural characteristics, biochemical characteristics, host range, virulence factors, and mechanisms of antibiotic resistance. Let's begin. Campylobacter jejuni is a bacterium that causes gastrointestinal illness. It is mainly spread through contaminated food, especially poultry and dairy products which have not gone through pasteurization. Good food safety and hygiene practices are essential to avoid exposure. C. Jejuni is a bacterium that is usually found in the intestines of animals including poultry, cattle, and swine. Additionally, it has the ability to thrive in the environment, for example in water sources and on contaminated surfaces. This slide discusses C. jejuni's habitat. C. Jejuni is a curved or spiral-shaped bacterium with a unique wing-like appearance, allowing it to colonize and invade the intestinal lining. Its possession of flagella also enables it to move quickly and efficiently, making it a challenge to remove from the body. When it comes to protecting oneself from the transmission of C. jejuni, it is important to be aware of the necessary precautions. C. Jejuni is a fastidious bacterium which necessitates particular, specialized media for its optimal growth. To ensure the most favorable results, the media must be devoid of any blood products. This microorganism thrives in microaerobic conditions, that is, conditions with reduced oxygen levels and augmented carbon dioxide levels. Campylobacter jejuni is a microaerophilic organism, meaning it thrives in environments with low levels of oxygen. It is also catalase and oxidase positive, showing the ability to produce catalase and oxidase enzymes. As it is a non-fermentative organism, it does not use carbohydrates to produce energy. Campylobacter jejuni is predominantly linked to poultry but is also capable of infecting different types of animals, such as mammals and birds. It is the most common cause of human gastroenteritis, which can cause discomfort and further health issues. The slide looks at the various virulence factors Campylobacter jejuni produces to cause disease. This includes adhesins, invasion proteins, the cytolethal distending toxin, and flagella. These components allow the bacterium to attach to and make its way into the intestine, produce an inflammatory response, and cause cell damage. Campylobacter jejuni has become increasingly resistant to antibiotics, especially fluoroquinolones and macrolides. Resistance can be acquired through the acquisition of new resistance genes or through mutation to the target sites of these antibiotics. This decreases the effectiveness of antibiotics, rendering them less dependable when treating infection. This slide presents an overview of Campylobacter jejuni, one of the most common causes of bacterial gastroenteritis around the world. It is estimated that millions of cases are reported each year and can be caused by the consumption of contaminated food, such as poultry, or exposure to contaminated water or animals. Campylobacter jejuni is a bacterium that, when ingested, colonizes the intestinal lining and causes an inflammatory response and damage to the epithelial cells, resulting in common symptoms such as diarrhea, abdominal pain, and fever. As these symptoms are similar to other foodborne illnesses, it is important to identify the presence of Campylobacter jejuni in order to properly diagnose and treat it. Our immune system responds to Campylobacter jejuni infections by producing antibodies, especially IgA, to inhibit bacterial colonization and counter toxins. Investigating these immune responses can help us create more effective methods to improve immunity against C. jejuni infections. The diagnosis of Campylobacter jejuni infection involves collecting stool samples and testing them with selective agar plates and molecular techniques such as PCR. Awareness of these methods is essential in order to ensure accurate diagnosis of C. jejuni infection for healthcare professionals. Preventive measures against Campylobacter jejuni infections are of great importance. Thorough cooking of poultry and avoiding of unpasteurized dairy products are essential safe food practices that can reduce the risk of transmission. Additionally, hand washing and other good hygiene practices should also be employed to better protect against this potentially dangerous infection. Campylobacter jejuni is a common bacterium that can cause foodborne infections, which may have serious effects. Generally, the disease is mild, but in certain risk groups or severe infections, antibiotics such as macrolides or fluoroquinolones can be used to manage it. It is important to follow medical advice to ensure positive results. Campylobacter jejuni is a gram-negative bacterium that affects humans through contaminated food. To effectively manage and control campylobacteriosis, it is important to understand its characteristics, epidemiology, pathogenesis, diagnosis, prevention, and treatment. Maintaining high hygiene standards in food consumption is essential in order to reduce the risk of exposure to Campylobacter jejuni.
Thank you for watching. I appreciate your support and hope you enjoyed the video. Don't forget to like, comment, and subscribe for more content.